let's see, of our chapter 16. Remember chapter 17, is all about transcription and translation, or about gene expression. How do we go from a gene to a protein product, right? So we're going to start here on chapter 17, part 2, the genetic code, and our superhero here says we're almost ready for translation. So now we're thinking about how does a code of nucleotides somehow get translated into specific amino acids? Because that's what happens in translation, right? Our nucleic acid RNA gets translated into amino acids. Does anybody remember how many different nucleotides are found in messenger RNA molecules? Okay, let's see if we can remember this. I will get my little pencil out. Did any of you say 4, right? There are A's, there are G's, there are C's, and there are T's. No T's. There are U's, right? How many different amino acids are in proteins? Ooh, this is something from chapter 5 material. How many different amino acids are used to build proteins? 20 different amino acids. You can look them up in chapter 5. And if you take biochemistry in the future, you'll have to memorize them all. Yay. So the question is, if we were using if we were translating the nucleotide code into amino acids, can one nucleotide code for each amino acid? Well, no, there are only 4 nucleotides and 20 amino acids. What about a 2 nucleotide code, like UU, AA, GC, AU? Can 2 nucleotides code for each amino acid? Are there even enough combinations of pairs of nucleotides? Well, you could actually sort that out by doing some math. There are 4 different nucleotides, and we want them in groups of 2. There are 16 total combinations of groups of 2 nucleotides, so that's not going to work very well. We have 20 amino acids we have to code for. So no for 1 nucleotide. What about a 3 nucleotide sequence? Can 3 nucleotides like AAG, AGC, AAA, could that work? How many different combinations are there? Well, there are 4 different nucleotides. If we group them in groups of 3, there are a possibility of 4 times 4 times 4, or 64 different possible 3 nucleotide codes, if you will. So it's possible. Is this how it works? Does a sequence of 3 or 3 nucleotides code for 1 amino acid? Yes is the answer. Here's a picture that I really like, actually. It's worth studying for a second, of DNA transcription and translation. And so at the top we see a DNA molecule, a piece of it, right? And there are 2 components to that molecule, right? 2 strands. One is the template strand that's going to be used to build the RNA molecule that complements the RNA molecule, and one is the non-template strand right there. They run in opposite directions. During transcription, the template strand is used to build an RNA molecule. And the RNA molecule runs opposite, so we got, our template strand running 3' to 5', our RNA molecule running in the opposite direction, and it should complement the template strand. So when there's an A, we put in a U. When there's a C, we put in a G. When there's a C, we put in a G. When there's an A, we put in a U. When there's an A, we put in a U. Okay. Now in translation, each triplet nucleotide sequence, they're called codons, codes for the building of 1 amino acid. So the codon UGG in the 5' to 3' direction codes for this amino acid TRP, which is tryptophan. The codon UUU codes for the amino acid phenylalanine. etc. Okay, the codon GGC codes for glycine, and the codon UCU codes for serine. So this is actually what happens. Each triplet nucleotide sequence, each codon codes for the building of one amino acid. Okay, this is easy. And in fact, researchers, after they discovered this, quickly compiled a dictionary of codons that lists all of the codons, all 64 possible codons, and the amino acid that each codon codes for. So we're going to take a look at these questions. What are some features of the genetic code? Is this code universal? Is there ambiguity in the code? So here's a picture of the genetic code, if you will. This is our dictionary that tells us what each triplet nucleotide sequence in RNA, which amino acid it codes for. And so let's look at this a second. The first, our mRNA base is here. You can either have a U, a C, an A, or a G. The second mRNA base is here, a U, a C, an A, or a G, and then the third base is here, UCAG, UCAG. Okay, so all of these codons, like UUU in the top corner, that's a codon, a triplet nucleotide sequence. There are 64 of them, if you count them all up. And they each code for amino acids. So UUU codes for the amino acid that's called PHE. PHE is just the abbreviation of phenylalanine. There are different abbreviations for all 20 amino acids. But notice that UUC codes for phenylalanine also. There are 2 codons that each code for a phenylalanine. A couple other things to notice about this is the start codon, AUG, says that it's methionine. It codes for methionine, that's an amino acid, but it is also the start codon. That's the codon where translation is going to start. And then over here, we've got 3 codons that don't code for any amino acid. They are stop codons. UAA, UAG, UGA. They don't code for any amino acid. They cause translation to stop. And let's see, what else do we want to ask? Is the code universal? Is there ambiguity in the code? Okay. Is this code universal? Could I use this dictionary to translate an mRNA molecule in humans, in bacteria, in fungi, in plants? Yes. For the most part, this code is universal. UUU codes for phenylalanine, whether it's in bacteria or humans. That's cool. Is there ambiguity in the code? In other words, if I ask you, hey, what does AAU code for? Can you always tell me? Well, you look it up. AAU codes for this thing, ASN. Maybe you don't know what that is, but it's not ambiguous, is it? No, we know what each codon codes for. It's redundant, though. In other words, there are sometimes more than one codon for a single amino acid. Like look at leucine. Leucine has 6 different codons that code for it. So there's some redundancy in the code, but it's not ambiguous. All right, so now we can use this code and we can translate anything we want. So let's do 2 or 3 problems, and then we'll look at how translation happens inside cells. But we can do any kind of translation we want to now. So here's a practice problem number 1. What polypeptide product would you expect from a poly-G mRNA that is 30 nucleotides long? So let me think. Poly-G means many G's in a row, 30 of them. 1, 2, 3, 1, 2, 3, 1, 2, 3. Oh boy, we're going to be doing this a lot, right? 30 of them long. 10 of those codons. Well, what does GGG code for? So we look it up. GGG. Here it is. GGG codes for glycine. So I think I would have, for each triplet, I think I would have glycine. How many of these would I get in a row? I would get 10. So my practice problem says, what polypeptide product would you expect? Well, I would expect glycine, 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 glycine 10 times. We just did it. Let's try problem 2. What polypeptide product, in other words, we're looking for amino acids, would you expect if this sequence of mRNA is translated? Now, translation always happens in the 5' to 3' direction on the mRNA. So there's the 5', there's the 3'. We're simply going to translate each nucleotide, each codon, or each triplet nucleotides into an amino acid. So, what does AUG code for? Well, that codes for methionine. So methionine is our first amino acid. What does CUU code for? Well, I have to look it up. CUU codes for leucine. What does UCU code for? Gotta look that one up too. UCU. Oh, it codes for something called SARE or serine. And what does GAG code for? GAA. Where's our G? GAG. You're down here. Codes for GLU, glutamic acid. Okay? There we go. We just made our polypeptide product. Oops. Oh, I don't know what I did. There we go. I can barely read my last amino acid there, but it's there. That's all we have to do. Make sure when you're using the start codon to report that that's methionine. That is methionine. Don't just write start. It's methionine. Okay. Let's do practice problem number 3. What is the sequence of the DNA template that corresponds to the above mRNA sequence? Oh, that's the sequence we just translated. And it says assume that there are no introns removed in the mRNA molecule. So the DNA molecule should just be, we should be able to find the DNA template pretty easily. The template just complements the mRNA molecule. But which direction does it run? Does it run the same way as mRNA, or is it anti-parallel? I think it's anti-parallel, okay? And then we simply, if mRNA has a U, the template DNA molecule must have has an A. A pairs with T. U pairs with, U is like a T, so it pairs with A. G pairs with C. C pairs with G. U pairs with, so that's like a T. It pairs with an A. Now we're going to get a bunch of A's here. 3 A's in a row. C pairs with G. U pairs with A again. G, oh, I'm not quite done. You have to write smaller than me. G pairs with C. A pairs with T. G pairs with C. Wow. There we go. I just made the DNA template that complements, and you can do this, that complements the RNA molecule. This is super easy. This is what happens inside the cell when translation happens. Okay, so now we have to figure out really, how does this happen inside the cell? How does translation occur? How do we go from an mRNA molecule to a polypeptide chain, a chain of amino acids? Let's just write that down so that we remember that. Okay, first of all, think about where this happens. Where does this happen? Where does translation happen inside a cell? It happens not in the nucleus. It happens in the cytoplasm. Okay, so we're sitting in the cytoplasm with an mRNA molecule, right? Here's our mRNA molecule. It's running 5' to 3'. It has a sequence of nucleotides on it, right? And it's been processed. It's now moved out of the nucleus, and now we're going to translate it. All right, we need some machinery to do the translation. We need tRNA molecules. That's one thing we need. tRNA molecules are called transfer RNA molecules, so that's different. We haven't seen that before. They are the adapter molecules, so let's write that down. Adapter I just saw my daughter running from our house across the street. There's a neighbor they're gone for like a month, and we're using their house. We're also getting their mail and stuff, but we're using their house as a second office. And my daughter just ran over to my husband. She must have some 5th grade problem that she's working on that's too hard for her. Okay, so the tRNA molecules are going to be our adapter molecules. They're pretty important because they see the RNA language, the nucleic acid language, and they translate it into the amino acid language. All right? So let's look at what tRNA molecules look like. I'm going to turn my notes to the right page. Oh, I have nothing here. There's another picture in the future. But a tRNA molecule is now does the job of translating. It's like got the hard job. And what it is an RNA molecule. It's single-stranded, but it folds up on itself, and it folds up so it kind of looks like kind of a clover shape, okay? Something like that, where it has a 5' end and a 3' end, and it kind of forms some pair bonds with itself in certain spots, so that's why it looks like it kind of folds over on itself, okay? And it has a couple of important features. One of them is, so it's a small RNA molecule, it's about 80 nucleotides long, and 2-dimensionally it looks like a clover. 3-dimensionally kind of looks a lot more interesting. Sometimes I will draw it more like this, and we'll see why in a second, okay? But it has, importantly, an anticodon loop. The anticodon loop is down at the end of the molecule. It's called the anticodon loop. Now it runs 3' to 5', and the anticodon loop is made up of 3 nucleotides that recognize and bind to a codon. The anticodon loop complements a codon on the mRNA molecule. So if we had an mRNA molecule, that's our guy down here, this is our messenger RNA molecule, and somewhere on this molecule is the start codon, AUG. An anticodon that recognizes this codon will have 3 nucleotides that complement AUG. So instead of an A, it will have a T. Oh wait, it's not a T. Oh, a U. Instead of a U, it will have an A. Instead of a G, it will have a C. So my tRNA molecule, in this case, its anticodon loop is UAG, and it complements with the codon AUG on an mRNA molecule. In addition to the anticodon loop, there is what's called an acceptor stem or an attachment site. Let's call it an attachment site for amino acids. At the 3' end of the molecule, an amino acid will attach itself to this tRNA molecule, and that amino acid is the amino acid that our anticodon loop. Now, let me see. This tRNA molecule recognizes the codon AUG. Its anticodon loop complements the codon AUG. It sees AUG and it translates AUG into methionine. So what it carries on its attachment site is methionine, the amino acid met, or methionine. Okay? So every tRNA molecule has an anticodon loop that recognizes a codon, and every tRNA molecule has an attachment site for an attachment to the appropriate amino acid that the anticodon loop, that the codon codes for. Okay? Let's see if we have, when we can draw this could be our anticodon loop. We can draw it more like a kind of a funny L with its amino acid at the attachment site at the end of that L. This would be our anticodon loop then, okay? Notice that the anticodon loop is running anti-parallel to the mRNA molecule. Here's some pictures of the two-dimensional shape of the mRNA. I'm sorry, the tRNA molecule. Sorry. And this is more of its three-dimensional shape. We like to use this kind of funny L shape just because it's easier to draw and easier to see. Well, you can see its anticodon loop here, okay? All right, so let's fill in a few of these pieces here because we've already covered them. The tRNA molecule is a small RNA molecule with a clover leaf shape. The anticodon loop recognizes